What are polymers? The way I've been looking at it is poly just means multiple, and I've got nothing for what mer means. Okay? So we're just looking at just a repeating unit. Something just repeats on and on and on and on. So if we looked at a polymer, we now have a repeating unit within each of these. Okay? And then the next part is that we can end up with different types of monomers in our possible polymer. Okay? So we could look at a situation where they aren't all the same. In this case, we got the red and the green to help kind of illustrate this. You'll notice that in one option, they alternate. And in the other option, they don't alternate. Okay? And theoretically, in that last one, it would be more random as far as what things we're connecting. There is no kind of repeating pattern within that. Okay? So we have multiple types of polymers that could come out of this. We want to come up with some way to reference them so we have some understanding of what's happening between them. So in the first one, or our very first one, the black spheres are just polymers. And we can not have to worry about coming up with some other name. If we have two different monomer subunits, we can reference these now as copolymers because you're polymerizing two different pieces, okay? At least two different pieces. And then in that last case, you have something referred to as a block copolymer, just meaning that we have a whole bunch of monomer units that repeat, and then they're broken up with repeats of other monomer units. Okay, there really is no affiliation per se on how many repeats within those. That's why we get these blocks or sections of those repeating units. The next big concept that can come out of this is this idea of cross-linked. Okay? So if we look back at this slide, you'll notice that within each of our monomers, I'm showing a stupid little black thing coming out the end of each of those. That black thing is where they link. Okay? To have a polymer, we have to have at least two linking spots so that it can bridge and continue to grow that structure outwards. If our monomer, for some reason, has more than two possible bonding sites, okay? So in this case, we've got one with 4 possible bonding sites. What we can end up with is a structure that is now cross-linked. Okay? And it doesn't have to be the entire polymer. We can have cross-linking molecules randomly inserted into our structure. Those cross-linking molecules can now link multiple chains with each other. Okay, for instance, we could have had one chain running through here and one chain through here. Those two chains are now linked to each other through a monomer connecting that bridge or connecting that gap. That's going to change the physical properties of our polymer. Okay, so depending on what we're trying to do, we may want cross-linking, we may not want cross-linking. Okay, how would cross-linking potentially affect our physical properties? What do you mean melting point? How? Yeah. Should increase it. You're now linking these things much, much tighter to each other. Not only is it now intermolecular forces between those polymers, they're now cross-linked to each other, meaning they are chemically bound. To get that species to melt, you're actually going to have to break bonds. So you're probably going to end up looking at a decomposition instead of a melting. Okay. Other things. More rigid. That structure is now stacked on top of itself and reinforced in multiple different directions. That tends to make a much more rigid polymer, not very free to move, not very elastic. Okay? So that cross-linking can change those physical properties and change ultimately what we want out of this. The part that you're probably most responsible for is a shorthand coming out of polymers. Okay? And this is largely because really when we look at the polymer chain, this isn't a very good representative polymer. That should run for probably 1,000 times, 2,000 times. To show that structure on this page would be tedious at best, okay? And at worst just completely meaningless because we're looking at too large of a structure. So what we want to do is kind of zoom in and change our focus to just look at the repeating unit. So what is the repeating unit in this case? and this is always fun, I heard a circle, so there's a repeating unit. I think I also heard someone say there's 2 circles as a repeating unit. Are both of those valid? Yeah, both of those are repeating units. Okay, so both of those are equally valid. Okay. Unfortunately, when it comes to finding polymers and patterns and naming things, it comes down to somebody saying, this is the way we work. And because of that, showing 2 does not count. In this case, we only want to specify that there is that one system or that one unit repeating. How many times is it repeated? Okay. Is it actually 8 times? I didn't actually count. Okay. If we looked and assumed that this was the end of our polymer, we've got 8 of those, so we could go through and say 8 in our lower right-hand corner, saying it's repeating 8 times. We've seen this before. Yeah, we've seen that shorthand before. It does show up. Okay. So that's all we're trying to represent with that notation. If we don't know how long the polymer is, sometimes you'll just see reference to an N. Or if we don't care how long it is, we just say it's N, it's some number. Typically it is an N equals 300 to 500. Okay. You'll be given a range. Why a range? Unfortunately, we don't have the tactile error control or quality control with building our polymers to get too tight of a range. Okay, so we just throw our chemicals into a soup and we let it react for a while and we stop the reaction at a certain point and we take a rough guess as to how much actually reacted based off the molecular weight of our compound, percent yields, all that fun stuff. Okay. What about these? What would our repeating unit or our shorthand be for these?
For the middle one, it would just be a circle and square.
Okay, for the middle one, circle and square. Anybody disagree? Oh, come on, be adventurous.
It could be circle, square, circle.
Why not square circle? Okay. And yes, you're looking at, well, this is stupid. Why should I have to worry about this? Unfortunately, again, same rules as the last one. One of these is correct, one of them is incorrect. Okay. How do you know? Okay. It goes back to polymer rules and saying this is the way it works. Okay. Typically, when you're assigned or trying to come up with the notation for polymers, you put the highest priority polymer or the highest priority atom first. In this case, it's kind of difficult to figure it out because they're shapes. We don't really have a higher priority shape in this case. But when we're looking at trying to divide up our polymers into our shorthand, that's what we're trying to do, is put our highest priority atom first. Okay. As if that wasn't complicated enough, what happens if your highest priority atom is in the middle of your monomer repeating unit? So for instance, if we looked at our circle up here, okay, that is how we sliced it. But what if your highest priority atom within that circle happens to be somewhere in the middle? I know this is a weird concept. Now if I copy what's in there and paste to the left and to the right, does it copy?
Yeah.
Yeah, it does. Okay. So there's a multitude of ways that we can go through and cut up our polymers to generate these repeating units. Depending on who you're talking to and what they are looking for, you may see some of these odd variations. Most commonly, what you will be looking for as your variation is the one that actually shows the repeating unit itself, intact, not being cut up and broken to pieces. Why? It's easier to see the repeating unit. Okay? The idea behind a shorthand is to make it more obvious, not less obvious, as to what's happening within the polymer system. Okay? So most commonly you'll end up seeing it grouped around an individual monomer unit. Okay? What about the last one? And the block copolymer. And as a hint, we'll probably actually have to draw it out separate.
You probably have to assume that it's going to be 3 circles and 3 squares.
Or not, unfortunately. Okay. This notation is saying that those do not necessarily alter, that they will go in blocks and they are somehow connected to each other. That is the notation you end up seeing in the block copolymer. Okay. As opposed to just the copolymer. The copolymer will show both monomers in your repeating unit. Okay. Questions about that? Okay. So take a second, see what you can come up with for your possible shorthand for each of these polymers. Okay. Do what you can. If you got questions, raise your hand. I'll make it easy to see what? The monomer. There is no way to generate a single carbon to polymerize. It's not going to happen. Your repeating unit is here. So a couple things to note. Notice that what I'm including within the parentheses are 2 carbons. I am not including the carbons on the outside. The parentheses are being drawn through the bond, not through an atom. So now what will happen is if I copy and paste what's in here over here, I get the exact same result. If I copy and paste to the left, nothing changes, everything matches exactly the same. This can become a bit challenging with the other examples because in the other examples they aren't particularly long, so you may not see that you mismatched somewhere through there. Okay. So take another swing at it and see what you can do. Number 2. How many of you came up with this as your repeating unit? Technically wrong. How many of you came up with this as your repeating unit? Also technically wrong. How about that one? How many come up with the one in red? I know there's lots of lines covering that. That one's actually right.
Why is that higher priority than the carbonyl?
Which atom is first in the blue one? It is a carbon versus nitrogen. Nitrogen takes priority over the carbon. Okay. Within shorthand, I'm fully aware that it is a complete nightmare to go through and find these. I will accept all 3 answers. The answer that I would hope that you would get to and that you would see first would be the one in blue. Why the one in blue? All of them were repeating patterns. I don't quite accept that. And there's a big hint. What kind of bond is that? Yes, keep going. Yes, it is also a sigma polar bond. Keep going. It is an amide, which is technically an acid derivative, which we just studied. You know how to make amide bonds. Okay. Why show the polymer breaking through that bond? That is the bond that is formed in the form or the process of making your polymer. So what we were trying to show with that grouping there is actually the monomer unit. Okay? It helps us see the monomer unit within this polymer. Okay? So we could then look at answer choices and say, well, that answer choice matches because that is my repeating unit. Okay? Or that is my monomer. It technically doesn't fall under IUPAC rules for assigning the priorities of your parenthesis cutting, if you will, and determining your shorthand notation. Number 3. How many of you gave me this? That's good because that one's straight up wrong. Okay, a couple people had this at first. We have to remember you're copying and pasting directly outside. So if I copy and paste out, that has to be a red carbon. The red carbon cannot have a methyl attached to it. So we drew the parentheses through the wrong bond. How many of you gave me that one? Okay. Does that show the repeating unit? Yes. Did it put the highest priority functional group or the highest priority atom first? Okay. The red one is technically correct. Okay. Within these, I will accept credit again for all of these possible cuts because they do count as valid ways of looking at it through varying extents, more correct or less correct. The red carbon, because the first carbon within that sequence has a higher priority than the first carbon in the other sequence because it has that extra methyl group. It was a so?
Yes. What if you were to draw the parentheses through either 2 blue carbons or 2 red carbons?
So we can't split atoms. That's why we don't draw the parentheses through the atoms. Yeah.
Just curious.
Last one. Is that a correct repeating unit? Kind of hard to see it because the chain didn't go out long enough, but that would work. Nitrogen's the far thing on the right. If we pick this structure up and move it over and drop it down, what do we have? Nitrogen. So that would work. You'll notice that this is a very long system. Okay, and we need to include all of those carbons. We can't start cutting small sections out. Because if I did that section, sure, nitrogen's the next atom. Okay, but what would be the very next atom? The carbonyl carbon, which is not present there. Kind of see it?
So what happens if you start at the first nitrogen and then cut it off?
Is there another correct way to show this one? The nitrogen has higher priority, so technically you should be cutting where the nitrogen comes first. And this is one that I'm not a particularly fan of, but still wrong.
Because you want the one that's going to be a carbon.
Yup. Okay, red is your IUPAC. What's the problem with red? It's cut up the individual monomer units that went into this. Okay. We're actually not putting the parentheses through the bonds formed. Okay. Where are the bonds formed in this case? Same place as they were in number 2. It's the amide bonds. So it's helpful to draw our parentheses through those bonds because we can better see the individual units. So what we've got within this slide is a big confusing mess of ways that you can come up with shorthand. And I'm fully aware that it is a big mess. It's usually devoted to an entire class, not a class period, an entire class, whole semester of material on polymers because there's a lot of information that goes into them. So what we are looking for is trying to find that repeating unit and starting to make some kind of generalizations about commonalities that we see within those repeating units. Can I erase everything that's up there? oops, I wanted this. Go ahead.
Can you tell us what the correct one was for
Number 2, your best answer is that. Why do I have these circled as such?
Because you want to give us any of the
What? I want to give you what?
You
Something?
want to give us any of the purple?
No. In fact, purple is what you're responsible for now. What's shown in purple is the process of making amide bonds. When we go through to make an amide bond, it was discovered when reacting, say if we pick that first one, when taking NH2, actually let me draw that out this way. What compound is that? All you aspiring biochemists? I think it's glycine. It's an amino acid. We're just looking at a protein in that case. What happens when I copy and paste the polymer out the other side? There's my amide bond. What was just scratched out? H2O. When these reactions were first discovered, when they went through and did them, what they ended up noticing was in the course of the reaction, not only were they exothermic, but it was exothermic enough that if they looked up on the glassware, they started to notice condensation. So the reaction is known as a condensation. These are condensation polymers. In the condensation polymer, you will always have a carbonyl and you will always have a good nucleophile. That nucleophile is typically a nitrogen or an oxygen. Okay. So by recognizing something about that structure, you now have a way to say, okay, I know what polymer I'm looking at. These are the monomer units that I need to use. Okay. The carbonyl can't be an aldehyde or a ketone. Why not? One option is can't make water.
No, so you can't do a condensation reaction.
All of your condensations result in the elimination of a small molecule. Can you eliminate hydrogen from a structure? No. H- is not a stable leaving group. How about a carbon in, say, of a ketone? Not a stable leaving group. So to do a condensation polymer, you have to be working with your acid derivatives. They're the only ones that can go through and do this chemistry. What did we do in chapter 20 and 21? Nucleophilic elimination from carbonyl compounds. It is a condensation polymer. It's just you're doing that chemistry now hundreds of thousands of times, okay, to make your polymer. If we take a look at the ones in red, you'll notice that you do not see any carbonyl chemistry. Okay? One thing that you want to watch out for, just to be tricky, we could potentially throw in, say, maybe a carbonyl out here. Does that now make that a condensation polymer? No. What you need to do is focus on the polymer backbone. The polymer backbone is all through here. Do we have any carbonyls in the polymer backbone? No. So it cannot be a condensation polymer. We are in fact forming bonds between carbons and other carbons. Where have we made those types of bonds before? What was that? Radical. Yep. Addition reactions. You're doing an addition reaction in these upper ones. Okay. The exact mechanism can be radical. That exact mechanism we'll talk about here briefly in a second. Okay? So two main categories: addition polymers, condensation polymers. Within each of these, what you're trying to do is look for those individual monomer units as your repeating unit to help you process what's happening within the structure. Okay. The very bottom one was actually an interesting condensation polymer. What is the monomer repeating unit? What bonds did we say needed to be made in the condensation? What is odd about what I have highlighted?
Is that a triglyceride?
There's an amide bond in the monomer backbone. Why? There are two monomers. This is a copolymer. One of those is the nitrogen with 6 carbons. The other one is the carbonyl compound with 4 carbons running through the middle. There are two monomers in this structure. When we mix those two monomers, they react to form the extended polymer structure. It is not the block copolymer because it's not nitrogen repeatings with nitrogens. It is always going to be a nitrogen to the carbonyl. Okay. So slightly different from our amino acid, because in our amino acid one, notice that your nucleophile was attached to the same monomer that our electrophile was. In the case of our other one, our nucleophile is not directly attached to our electrophilic molecule. Okay? Within the shorthand, you don't show that. So how would you end up showing it? Well, the question would be, what are the correct possible monomers for this structure? Okay. What they would end up showing is two molecules. One, the diamine, the other one, the dicarbonyl compound. Okay. This last compound, I would guess almost everybody has on their person right now. It's nylon. That is the structure of nylon, which we'll be making at the end of the semester. When we look at polymer formations, your addition polymers, right? You're starting with an alkene, you react that with another alkene, and that goes through and propagates the structure outwards. Typically, you're looking at these as free radical reactions, but you can also see them as cation or cationic or anionic polymerizations as well. We're using a negatively charged species or a positively charged species to initiate the reaction. Some things to watch out for: electron donating groups and electron withdrawing groups can mess with the arrangement of those things. You'll notice that we always saw alternating methyl groups. Why did we see alternating methyl groups? The methyl group acts as an electron donating group, which means there's now a buildup of electron density at that position. I would not expect this polymer or this monomer to attack its mirror image. It needs to be aligned head to tail. Kind of make sense? Within the condensation polymers, what you're doing is that elimination reaction within your carbonyls to eliminate that small molecule and end up with your polymer backbone. When we look at our addition polymers, most commonly you're looking at these as free radicals. So you add in some radical initiator, which causes a radical alkene. That radical then continues to propagate and react outwards to make your larger polymer structure. Within that, you've got your start of your polymer formation, which is known as the, oh, never mind, it's already up there, initiation. As the reaction continues on its own, it is the propagation. When the whole reaction is done, we have the termination. How do you know you're looking at an initiation step? In a propagation, you break a bond too. In the initiation, you create a radical. You do not start with a radical, you end with a radical. Propagation, start with a radical, and you get a different radical the end of the propagation. Okay? And your termination? Start with a radical, end with no radical. Okay? It's the same general setup as you've already seen when we looked at radical reactions way back when. Condensation polymers are a little bit trickier. What happens with our condensation polymer? Don't all shout it out at once. Go ahead.
The addition or elimination with another element solid.
So we're doing our addition elimination. That's our new bond. There's our nitrogen. Now what happens? Base comes in, removes a hydrogen. Now what happens? If we go to form the double bond, what's our good leaving group? OH is a good leaving group? Okay, since we'll run on that assumption, we're running in a basic environment, strongly basic environment, and eliminate that out. We'd have our polymer. Particularly if you turned it sideways, that helps you see it. Okay? This can now continue that process out either end. Would we expect this reaction to work very well? What was our good leaving group? What was our good leaving group? Maybe the silence is because there wasn't a good leaving group. Okay, we had hydroxide, and you just kind of say, well, it's because it's a basic environment, which is kind of a cop-out because we started with a carboxylic acid. That should be deprotonated. Basic environment is going to be a bad option. Okay. How could we make this reaction go faster? We could protonate, change our mechanism around, change those protonation states to help it work. What else could we do? How about instead of being a carboxylic acid, we start with ester is not going to speed it up. The ester is almost equivalent in reactivity to the carboxylic acid. Carboxylate is even less reactive. An acyl chloride, okay, or an acid chloride. Okay. How could we get that species to become an acid chloride? Could add SOCl2. What will SOCl2 do? Into our chloride. Will we ever be able to isolate this species? How reactive are acyl chlorides? Very, particularly in the presence of, try again, a really good nucleophile. Okay. So all we would probably have to do is add the SOCl2, and we'd probably get a very violent, uncontrollable polymerization reaction going off on this. Okay. How does your body go through and control this reaction? This is what enzymes were designed to do. Okay. Very carefully deal with the proton transfers back and forth so that we don't over-acidify, say, our blood or our cells, but facilitate the hydrogen transfers back and forth across the structure to help build a protein structure. Kind of a neat little process. Anionic and cationic polymerizations, this is more just as a factual thing. When we go through and extend our chain, if we take a look at that bottom example, this is an example of an anionic chain growth polymerization because we are using an anion to initiate the reaction. Okay. The reason we can call this an anionic reaction is because we end up forming a new stable anion. If we ended up forming an unstable anion, would it be able to do anionic chain growth? No. Okay, so it's all about making a more stable anion. If you do, you can do anionic step growth with this. Okay? Same idea with cationic. Crystalline solids versus amorphous solids. Polymers tend to be amorphous, which makes them a bit of a pain to categorize. We can't take a melting point to identify them. Running an IR can be a bit questionable as well as far as what information we can get out of it, because all of the bonds in our monomer look an awful lot like the bonds in our polymer. Okay, so we run into kind of odd results out of this. In standard solids, we've got crystalline, nice, organized, compact structures. When you move up to polymers, they tend to be so large that it prevents organized packing. Is this a good thing for us? Our entire system, living system, is based off of proteins and DNA. If those packed nice and organized within our blood system, that probably wouldn't work so well as far as mobility. Okay? So having these amorphous solids allows them to change their shape and structure under different conditions and gets better properties out of it. Okay? Which then brings us to this kind of transition. In a crystalline solid, we can refer to the point at which it turns into a liquid, or your melting point. When we look at an amorphous solid, we don't really have a melting point. Okay? We can kind of get one, but not very well. It just starts, it's the point at which your substance or your polymer just kind of starts to sort of melt. Hold some of its structure, but not entirely. This is known as your glass point, okay, or your glass transition. Questions on polymers? Okay, very quick whirlwind through on that. So what you are responsible for being able to do is to recognize those shorthands, be able to interpret them, and hopefully be able to find monomer repeats out of those. Okay. Have you already been tested on this? Yes. Go back, look at exam 1. There's an addition polymer in exam 1. Okay, so you will see a condensation polymer in this exam. Do you have any general questions? I'll pause the video. We'll try and go through it the way I would visualize it. It's a little bit unfair because I think I've done these questions now multiple times. I see the R isomer statement there and already panicked and moved on. Using knowledge of base strength, I'm serious, it's what I would have done. Base strengths, I would go through and then say, okay, you're asking me about bases, ignoring the rest of the question, negative to negative, which is the stronger base?
First. Or no.
The right is the stronger base, which would suggest what about the direction of the reaction? Favors the reactants. Now that I've done that quick interpretation, I might actually read the rest of the question, which says which of these reactions would be most likely to occur. Well, good. My little bit of work worked out. That first one should not work. If I move to the next one, I'm looking at hydroxide versus chloride. Stronger base. The hydroxide is the stronger base. Also not likely to occur. Now I would run into an issue. Because I would look at C, and I'd see negatives, and well, I've been looking at a bunch of negatives already. I'd look at D, and D has no negatives. So I would think, oh, well, D's more stable. You've got to be a little bit careful, ignore the rest of the questions. You're comparing these two species to each other. NH3 to SH, that has nothing to do with negatives anymore. You're looking at the base strength of each of those individual species. Which is the stronger base? Nitrogen is a stronger base. If nitrogen is a stronger base, the reaction favors the reactants. Okay? When you panic about that one, could be evaluated in the next statement. If we take a look at hydroxide versus phenoxide, which is the stronger base? That's not hydroxide, sorry. Methoxide. Methoxide is the stronger base because resonance. Resonance stabilizes the phenoxide ion, making that less basic than your starting material. C is your best answer there. Okay. If you didn't know between sulfur and nitrogen, I would at very least say the phenoxide is more stable than methoxide. C is absolutely favoring the products. Whether or not D does could be come up to a toss-up. Say, okay, well, I don't know. I'm just going to ignore it because I know C is valid. Does that make sense? Intermediate involved in base. As soon as I see base, what do I write? Negatives. I want to be seeing negatives in my reaction. If I see anything positive, that's not a good sign. And actually, just looking at this, I see positives everywhere. Positive, positive, positive. C is the best answer. What's going on with A and B?
It's showing,
In A, you're breaking the oxygen-carbon bond. That is not the most polar bond, which means it is not the weakest, which means don't touch it. A can't possibly be right. B is a little bit better. We are at least breaking the weaker bond there, but we're forming that positive charge again. Okay. Not a particularly good thing to do. What's going on in D? Someone can explain that one to me. I don't understand what's going on with D. Oh, it's just resonance. I don't know why I didn't see that. It's the resonance structure for the acid catalyzed intermediate. Okay. Acid catalyzed, again, not a good option. Okay. For which monomers are needed to produce this polymer? It's a good thing we just had a lecture on this. I know you've had so much time to practice it, right? I'd go through and identify the pieces. My weakest bonds for sure have to be on the outside. Okay. If I go through and look at those connections, I'm looking at some kind of polyester bond, so I would look within the structure and make sure that I don't have a polyester bond also showing up in the middle. I do have one in the middle, so I'm looking at two individual pieces. I need the pieces that match those as best as possible. The only one that matches those individual pieces is D. See the breakdown? 5. Which structure is a reasonable intermediate? Back to these stupid intermediate things. Now I'm looking at acid catalyzed. As soon as I see acid, I think positive. Go through and look for positives. There's a negative. That one's, and now I can only eliminate one answer, so now I'd have to evaluate a little more critically. Can I get a positive oxygen all by itself? We've seen positive oxygens before, which are reasonable in an acid catalyzed mechanism if they are protonated. Okay. A positive oxygen sitting out there like that does not satisfy the octet. So we have an octet issue. That structure doesn't work. Last one, the only way that would happen is if I just broke the piece off, this whole section off on its own. Okay. There's no reason for that to just leave on its own. Something has to help it off. So D doesn't make sense. Fun thing about all this, technically this is a nomenclature question. You should have also drawn out ethyl acetate. There's your acetate, there's your ethyl. Okay. Did you need to draw it out? Context of this question, no. We could cheat and say acid catalyzed positive, do the best to eliminate answers. I didn't even have to go through and look at their starting structure. Okay. That is likely because all of the answers are going to use those carbons. If those answers did not use that, they'd be asking me a different question. Make sense? So sometimes you can just jump straight to those answers. I've prolonged and skipped my pain as much as possible.
I have a question actually
Yes.
about number 5.
Go for it.
Is it safe, because it's asking for a reasonable intermediate,
Mm-hmm.
is it reasonable to assume you're going to have, to look for 4 different bonds, or like?
Good point, actually. Yeah, when we look at
That's what I thought.
our nucleophilic addition and elimination, the whole process, one of the big things we ended up looking at was the, am I spelling that right?
Can you lower your?
Maybe I shouldn't lower it first. Is it right? Okay, I'll keep it lowered. You have a tetrahedral intermediate, so in your addition elimination reactions, you will always have a tetrahedral intermediate. So it's not a bad idea to look for that. Okay, you do see that answer in C as well, but C has that weird, well, actually you see it with A too. Yeah,
But then you look to ask.
yeah. So both of those contexts would have worked.
Because that's not eliminated.
D.
Skin right away.
R isomer, this gets a bit odd because when we look at the structure, is there any hint that that is the R isomer? Is there even a hint that that has a stereocenter? No. Okay, so what I would do is mark what they're talking about. Find the stereocenter. It's that carbon. Now that I know what carbon I'm referencing, now I can look at the rest of the reaction. The reaction is looking at, again, a nucleophilic addition and elimination reaction, which means all the chemistry happens where? At the carbonyl carbon. That's where all my chemistry occurs. Does any of that chemistry affect the blue stereocenter? No. Which means B and D cannot be correct. Okay, because those show the S isomer. All of my chemistry is happening at a different position. It's not going to affect the stereocenter. Okay. What's our difference between A and C? It's the location of that weird O18. I've had a lot of people complain, what is 18 oxygen? Think back to 151, maybe even 152. It's an isotope. That's all it is. Why are they using an isotope? So that we can mark and follow how the mechanism worked. That's why it was in there. Okay? And it also makes it really easy to ask this question. Where did that atom go? In the course of the reaction, what happens? Your hydroxide comes in and we kick out the leaving group. So the O18 should be attached to my larger structure. There's answer C. Make sense? Fun thing to note on all of these. You'll notice how many of these just have an answer. None of them. Every single question has scribbles and marks and things helping to solve the question. Just because it's multiple choice doesn't mean you shouldn't write anything. Write your work down. Okay. Even some of the simplest markings, which I did for both 3 and 5. Base meant negative. Acid meant positive. Identifying something that I can look for, particularly if I have to come back to the question. Okay. Yes, this gets tricky on the final because on the final you're not allowed to write on the exam. There will be, what is it? Tracing paper, thank you. Okay, so you can write on the tracing paper.